This is a special episode of the Immunology Podcast, Immunology 2023, Day 4. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. Today we are back with our fourth special episode from Immunology 2023. As always, we'll be discussing our favorite sessions throughout the past 24 hours of the meeting. So if you weren't able to attend, we've got you covered. We're going to kick off things in just a minute, but before we get to that,
looking for a quick reference that you can hang on your lab wall? STEMCELL Technologies has various wall charts covering different immunology topics, including a snapshot of COVID-19, an overview of antigen processing and presentation, and more. Explore all the immunology wall charts and order your free copy at stemcell.com/immunology wall chart.
all right. Well, it's our last day here, a full day. We have a half day tomorrow, and a big day, covered a bunch.
What a day. Also, we had some recordings today. We got to see a lot of people and talk to a lot of people.
Yeah, so that'll be an episode coming out in June sometime, I think. And then we get the gala tonight, too.
So, Jason, what was the highlights of the scientific sessions today for you?
All right, well, they're getting more condensed from day to day. We seem to be in a concentration gradient here. So I went to Major Symposia F on myeloid cells, and there were a couple really cool talks. There was Jessica Hammerman talked about a TLR7 overexpression mouse and presented some previous data that it had enlarged spleens and increased glomerular nephritis. And then described really as a next step that they've been identifying that there's these splenic hemophagocytes that are coming up here that are very unique to this IL-7 overexpression. And they have linked this to juvenile arthritis with this spontaneous macrophage activation syndrome and seeing it there. And so that's becoming a really interesting model for that. And they're starting to get some therapeutic targets from it. In particular, IRF5 deficiency seems to protect from this. So that's kind of where they're starting to go down. So more to come, but it was cool to see. Ping Loke then talked about how hemolyths protect from SARS-CoV-2 infection. And so one of the interesting things in the pandemic is Africa never had the death rates that we're seeing in Europe and the US and other parts of the world, and wondering why. Was this underreporting? But it doesn't actually look to be that way, at least not all of it. And what they found is that hemolymph infections, so hookworms, things like that, can infect via T-cell, have a protective effect via T-cell activation, leading to macrophage activation. So what they see is that in the case where you have a hemolymph infection, they did this in mice and correlate with human data, that you have more CD8 positive T cells, and these lead to an alternatively activated macrophage at day 28, persistently out afterwards. And this macrophage type is better at killing infectious respiratory viruses. And in particular, they were able to demonstrate that alveolar macrophages are required for this effect using an intranasal ablation process where they did intranasal siRNA and really knocked out that compartment of macrophages. What was interesting is they talked about how they originally started with the wrong hemolymph model in mice, and one that only stayed in the intestine, and that didn't have the lung component, and they had to move to one that had the similar, intestine to lung life cycle to get the effect in the lung that was protective. So that was neat, just from like a model organism story. And then Paul Cubes in this talk had a talk about how they're looking at resident macrophages. And he was really tracking a couple different things. So once you establish that macrophages in the lungs can move across alveoli, and that wasn't known before, so he had some really cool videos of that. And also showing how monocytes can move into inflammatory areas to become macrophages. And he described this happens in large vessels during fibrosis, liver fibrosis, and then these clusters look like giant cells, and they probably are giant cells surrounded by stellites. But this is kind of an importance of how giant cells are formed. And then this last part of his story was that there's these Kupffer cells in the liver, right? These macrophage-like cells. And there's an importance in their location near the sinusoid. And so one interesting thing is in babies, the livers don't work the same. And he was able to show that E. coli catching in liver newborns is poor, but it gets better over time. And he's able to actually link that directly to the Kupffer cells migrating and establishing residence in the sinusoids of the liver. So those are the main talks. And then I got to get some papers.
Poster.
Almost the same thing. Differentially expressed, right? Different level of terminal differentiation.
Yeah.
But that being said, the poster I saw that was really cool is all these microbiome studies where, hey, this bacteria is good, this bacteria is bad in this condition. And we know that vancomycin ablation of bacteria leads to like a hyperinflammatory state due to decreased Bacteroides as one of the two major species of gram-positives that it'll ablate. And this paper linked actually that there's a specific bacterial lipoprotein or lipoglycoside, really. It's kind of a complex molecule, but it's bacterial metabolite that actually is what activates cells and decreases, so its presence decreases TLR signaling and TNF tone. And so you can give the mice the Vanco, and if you give this instead, you can keep them on the Vanco and it recapitulates. They stop looking like Vanco-fed mice. So it's recapitulating the effects of the microbiome without the microbiome being present by identifying some of these metabolites that have these signaling effects on our body. So it's really cool to see that. This has started to happen more and more in the field, but really being able to identify key molecules that the microbiome are acting through, right? Sometimes it's stuff on the surface of them, but sometimes it's metabolites. And so it's neat pharmacology to see. So that was my day mostly, besides all our awesome recording we did.
Yeah. All right. Very interesting. So I went first to the major symposium E, Engineering at the Interface of Immunology and Immunotherapy. And I like the talk from Jeffrey Hubble from the University of Chicago, in which he was talking about engineering cytokines to modulate region-specific function. I think the general idea was quite simple, and is binding cytokines to small molecules or proteins to just affect their location and where they are homing and how that is useful. And so the idea is how can we, for example, can we make tumors hot? And you can do that through targeting certain cytokines. How can we retain effector cytokines in tumors? And he used an example of collagen binding domains, which, as the name suggests, kind of co-localize with collagen and bind collagen. And so he uses that for example, doing an IL-12 therapy that will have a IL-2 fused to this collagen binding domain, CBD. And he shows that by doing this fusion protein, it can really direct the IL-12 towards the tumor specifically, or kind of increase and more enriched in the tumor. And that this actually does, this cytokine is functional and is inducing interferon gamma expression in the tumor microenvironment. And this really helps enhance the rejection of melanoma tumors in mice, while really having minimal systemic effects, which is the issue with IL-12, is really good for inducing immune response, but then you have the whole systemic effects can be quite crippling for the therapy. And he also shows that you can combine this with checkpoint inhibition, and you can make a cold tumor hot by using the CBD fused IL-12. And again, the toxicity, the systemic toxicity is reduced, and you see reduced levels of effector cytokines in the serum, showing that kind of all the action happens in the tumor. And he also gives other examples, for example, by binding to serum albumin. So, for example, if you do this with IL-10, you can increase the lifespan of the cytokine. And then he gives the example also of IL-33, which binds to a receptor called ST2 that is expressed in FOXP3 positive cells in Tregs. And he shows that by having IL-33 that is bound to fuse to serum albumin, he can treat Tregs and use them to protect against a model of MS in mice, EAE, and that this, despite IL-33 sometimes being associated with effector function, he can kind of, by using low doses, specifically targeted Tregs, and actually reduces leukocyte infiltration in spinal cord and reduces the kind of the development of EAE in mice, and also can also be helpful if if you use this EAE model, you induce it by MOG, by vaccination with a MOG peptide, so to induce a response against MOG, and it also helps kind of the chronic phase of the disease. So very interesting. Then there was another talk by Evan Scott at Northwestern University, in which he was engineering synthetic nanocarriers for targeted immune modulation. And I have to say, it was a lot of chemistry and a lot of, but it's very interesting. He really pushed the idea of using fully synthetic nanocarriers. So, synthetic polymers that like lipids would have a hydrophilic and a hydrophobic. So these lipids that are used for making like lipid particles for transporting small molecules or peptides or whatever. And so he shows that by choosing carefully the kind of synthetic polymers that are used to make these particles, he can really target specific areas in the mouse, and he can really he tries to use this in order to target dendritic cells in kind of a vaccination strategy. And so he has this particular system, which is a PEG PPS system, in which we had a polyethylene glycol, so PEG, which is hydrophilic, and a PPS, polypropylene sulfide, which is hydrophobic. And he shows that by tweaking the proportions between the two, he can make all types of structures, like of different level of complexity. He can make and one of the things he makes that is kind of interesting is a tubular structure that has the payload inside, and then as it degrades, it releases like little micelles with the payload inside, and that you can kind of pace the release of the active compound that's inside this polymeric structure. And he tries this, for example, examples that he gives is to target antiparasite compounds for Chagas disease. Another thing that he talks about is using decoy particles to because one of the things, if you have liver or certain macrophages, they're very keen on taking up these particles. So he suggests that if you do a treatment with a decoy particle, and then you fill up and you put inside these particles compounds that reduce pinocytosis by this target cells, then you can do a second round with your actual real compound, and then all these cells are going to be kind of taken and will not remove your particle of interest from circulation, and allowing it to get to the actual site of action. So basically, it was a lot of examples like this. Very interesting how you can choose different synthetic polymers to target compounds and peptides and antigens to different kinds of cells. And after was also the talk from Jennifer Elisif from Johns Hopkins, and she was also talking about immune stromal crosstalk, and it was a very complex talk, but I think the takeaway was that she was looking into healing after tissue injury, and particularly when you have implants or some kind of implanted material, and that often the wound healing around this material can be improper, and you get scarring, you don't get proper healing. And he shows that there's this includes senescent cells that are kind of induced by the production of IL-17 by Th17 cells, and that IL-17, by blocking IL-17, this seems to be a way to reduce improper wound healing in the case of implants. And I also went then for the talks from the American Association of Transplantation. They had a symposium too, and I was very interesting about the talk talking about the regulation of CD8 T cells by FGL2, and this was Mandy Ford at Emory University, and it was very interesting because she was looking into a particular receptor, Fc gamma receptor 2b, which she shows that is expressed in CD8 cells that are mediating allograft rejection. And she shows that this receptor is actually as a inhibitory receptor that has an ITIM domain, and that expression of this receptor on CD8 T cells is associated with a lower risk of rejection. And she does a lot of, work around understanding the role and how this receptor mediate this difference. And he shows that the activation of Fc gamma receptor 2b in CD8 cells, she has an ova-expressing skin transplant, and then she injects OT1 cells, and then they respond to the allograft. And she shows that the expression of Fc gamma receptor 2b on these cells is associated with an increased active caspase. And so they kind of basically go apoptotic. So the question is, what is the ligand? So what is activating this receptor? And it seems to be there's a suppressive cytokine, FGL2, which binds to this receptor, which can be secreted by FOXP3 so by regulatory T cells. So they first look into that. Doesn't seem to be the regulatory T cells. So in the end, she shows that it's actually activated T cells themselves that are making this FGL2, and that they are kind of mediating their own activation. So very interesting as well. So basically, she has FGL2 is released from the CD8 T cells, and it binds the Fc gamma receptor 2b, and this initiates apoptosis in the CD8 cells. Another interesting talk shows the effect of interferon beta, and so ways of modulating Treg function. This was from Paolo Cravetti at Mount Sinai. He was looking at inducing regulatory T cells for allograft survival. And one of the things he shows is that interferon beta that has been used to treat MS patients for a while. It is associated with increased frequency of circulating Tregs, and basically he shows that interferon beta induces an immunosuppressive program in Tregs and supports Treg function, and in particular the acetylation of FOXP3 in naive CD4 cells, and therefore the induction of Treg differentiation, and this can have a beneficial effect on reducing allograft rejection through the action of Tregs, for example, in the case of a heart transplant model. And he also mentions other molecules are known to help Treg function. I was surprised by erythropoietin, which I wasn't completely aware, but he also shows that besides being important for anemia treatment, which is the main use of EPO, it actually also promotes dendritic cells to induce Treg differentiation through production of TGF-beta by these dendritic cells. And yeah, so there was one last Treg talk. I'm going to just briefly say the takeaway message, looking into PPR delta receptors. The author was Qijian Yang from the Salk Institute, and he basically showed that PPR delta in Tregs is inhibiting the expression of MHC class 2 on the surface of Tregs, and that this negatively affects the function of regulatory T cells. And on the other hand, interferon, type 1 interferons, another way in which apparently they are supporting Treg function is by inducing C2TA and therefore MHC class 2 expression. And that this is a mechanism by which Tregs, they are in suppressing conventional T cells through kind of interacting with conventional T cells, and then, for example, through suppressive cytokine production, reducing their effects. So another mechanism for Tregs to inhibit T conventional activation. So yeah, it's a long day, a lot of stuff.
I am not surprised you ended on Tregs.
Yeah, well, you gotta end on a high note.
Well, with that brings us end of today's Immunology 2023 episode. Don't forget to follow us on Twitter at IMMUNO Podcast to find out what we're up to at the meeting. And we'll be back tomorrow after the half day of the meeting to cover the final bit of it on the podcast here. So check back tomorrow. See you then.